Jordan did his PhD here at ASU in the biological design program, probably the second batch, right, from the program in 2015 with Professor D.D. Meldrum. I remember presenting in Professor D.D. Meldrum's group, and there was the most incisive question that came from the back was Jordan's. And he then went on to do a postdoc with Dr. Vijay Singh at Mayo Clinic here, and then came back to do a postdoc at Biodesign with Dr. Alex Lucas. He then moved as research assistant professor to my lab in 2020, and then has done some amazing things before and since then. For example, he won the KO1 award from NIBIB NIH, the Career Development Award. The NIH is equivalent of, I guess, a career award. We won the Wound Healing Foundation 3M Fellowship from the Wound Healing Foundation and had to give an award lecture, which he did an amazing job with. Has won the ABRC New Investigator Award. That was, I think, in 2023. So he's done some fantastic things, doing some great science along the way. And then as we formed our center and we looked nationwide, Jordan applied to an open search and started as an assistant professor here in 2024. And already has now, in a new area, gotten NIH R01, the Stephen I. Katz R01, which promotes new investigators to completely look into new areas of research. He's starting his research group, has bought some fantastic equipment, and has reached out across centers for collaboration now, and is a great part of our center. He does some amazing other things, like being on the editorial board of PLOS ONE, and also on the communications, I think he's chair of the communications committee at the Wound Healing Society. So lots of activity, both in research, teaching, and service to both our institution, the Biodesign Institute, and also to the scientific community at large. It's my pleasure and also an honor to introduce Jordan Yaron, our 7 Minutes of Science speaker. Jordan?
Morning, everyone. Thank you so much for the introduction, Kaushal, and thank you for the invitation to present our work. In 7 minutes, I'm going to try to summarize work that even predates me in the center that's been going on for about 8 years, and then showing how it kind of evolved into what we're doing now. So the name of the talk is Biomaterial Tissue Crosstalk Reveals the Pathways of Healing. And I'm going to try to show you how our work in biomaterials engineering and investigating how these biomaterials interact with our tissues can inform us and educate us on how life works, how tissues respond to different conditions. But first, so I don't forget and so I don't run out of time, a lot of people have been involved in this work, both in the Reggae Lab, and so here I am in both pictures. I'm not trying to double tap my own shoulder, but so this is work that started when I was a research assistant professor with Kaushal, and then now I have the privilege of working with a lot of really talented young investigators as a mentor, and then of course we've been privileged to have a substantial amount of support over the past few years on this work. So diving in on the primary core question of what I'm interested in and what has percolated throughout many projects in the center is how do we restore full structure and function after injury? So if you think about healthy tissue function upon injury, we're all familiar with the fact that the immune system is recruited, stimulating inflammation that initiates reparative tissue responses. Over the course of a couple of days, sometimes weeks or months, that results in the recovery of different barriers, matrices, the induction of angiogenesis, and ultimately into a tissue remodeling phase that can last months or years, with the hope that we can restore healthy tissue function. But I'm sure everyone in this room at some part of their body has a scar, and that sort of percolates throughout many tissues upon injury, and that is evidence of our inability to close this loop. So if we can close this loop, we essentially reach a regenerative state. And so we are curious about what happens along the way and how can we manipulate these different processes in order to get to that healthy tissue function. So dermal wounds are a primary interest of mine, and this is an unusual thing for many people to think about because you think of dermal wounds as being something like a paper cut. But it may surprise you to know that while there are 6 million wounds per year, the cost is dramatically high, 20 billion annually in the US, accounting for about 5% of the total Medicare and Medicaid budget. And this is not simple paper cuts. These are wounds that affect people with diabetes, obviously burns and infection. Of course, advanced age can have complications with wound healing. 1 in 38 adults walking around the US right now is suffering from a chronic wound of some sort. And the total mortality of a chronic wound is along the same order as the total mortality of all cancers as an average. So these are things that are much greater in terms of medical burden than just simply slapping a bandage on tissue. So we look at this in a number of different ways, and I'll just briefly run through the core thinking that happens when you design a biomaterial, such as, do you want it to be natural or synthetic? And so this dictates how does something degrade or how long it should last. Do we want it tough and durable, or do we want it to be injectable? Should it be immune stimulating or immune silent? Do we want to put a drug in it? Do we want to deliver something within the biomaterial itself? And then something that I'm really interested in at my core is how do these biomaterials interact with cells and tissues? Should cells go into the biomaterial, and should the biomaterial act as sort of a scaffold? Should the material be cell instructive? And so do cells have certain responses to these things? So Kaushal has, for many years, developed a really rich research program in understanding how silk can be manipulated for biomaterial scaffolds. And so this is where I came on board to the team in 2020. So silkworms make these nice, beautiful cocoons where we can isolate silk fibroin out of. These fibers are composed of really 2 proteins: sericin, which is a sticky protein that coats a structural core protein of fibroin. So this is what you think of as like a silk is stronger than steel. It's usually this fibroin you're talking about. And so work coming out of Kaushal's group several years before I joined looked at how these biomaterials can be augmented with things like gold nanoparticles to make them photothermally labile. And so we can essentially make instantaneous scabs on any tissue with just the incidence of some near-infrared light. And so we designed a study looking at how we can take those silk-based biomaterials, modulate them with endogenous mediators of healing like histamine. So if you get a paper cut, it's going to itch. Next time you get a cut, think about it and you'll feel it itching. That's because histamine is released by mast cells around the injury to induce all of these behaviors. And so we took skin wounds and we had applied histamine or just saline, and we added either a clinical dressing called Tegaderm or silk biomaterials, added a splint, and then monitored the healing. And to make a very long story short, we see that different combinations of these treatments result in much accelerated healing versus just clinical dressing alone, as well as mechanical recovery. So I'm not going to talk more about this study except to say you can go read our work in Biomaterials from last year, and to also mention that the NIH was so excited by this that they wrote a little news article about it, because we really pointed to the fact that you need the right drug at the right time in order to get the best healing. When we did the transcriptomics on this study, some interesting things came out, including some evidence that we're manipulating a process called epithelialization. So this is describing how cells at the edge of a wound must get up and go into the wound space in order to close that tissue. And most people in the room, even if you're not a wound healing biologist, are familiar with this process because it's essentially the same thing that happens in cancers that leads to metastasis. So cells within a tumor will get up and go by undergoing something called epithelial to mesenchymal transition. And this is something that is absolutely essential for proper wound healing. So when we look at our transcriptomics, it turns out that one protein family that I'm particularly interested in lit up. These are called serpins. I'm going to give you a 10-second introduction to say that serpins are found in every branch of the tree of life. They act as sort of molecular mouse traps where they can entrap a protein by closing a loop, into a beta strand upon cleavage by a protease. But they can also act non-canonically to interact with proteins and act as chaperones. And so this is a core family of proteins that I've been studying for many years, even through my second postdoc. And we have a lot of serpins. So these are sort of the street names of these different serpins, but we now call them by their clade names, so serpin A1, A2, and so on. And you can see that these serpins have, these are all of our human serpins, they have really broad roles in cancer, which you can see here with the ribbon, but also there's evidence that they may have roles in wound healing. And in particular, this one was very interesting to me, the serpin B3 and B4, because this serpin B3 in particular, has been known for 40 years as the squamous cell carcinoma antigen. And it's so prevalent, in fact, in its importance in cancer that a patient with squamous cancers right now can go to Mayo Clinic, give some blood, and have a diagnostic test performed to see how much of this protein is circulating in the blood, and that's used to stratify the severity of some squamous cancers. However, despite 40 years of investigation into this protein, nobody knew why we made it. What is it for? What is the endogenous physiologic role of this protein? And so when you see, for example, high SCCA is associated with worse survival, and this is a squamous cancer and cervical cancer, showing just how much of that protein is there. So when we did our transcriptomic studies, you can see that the clinical dressing is showing some serpins light up upon wound healing, but this one serpin in specific, serpin B3A, is even more lit up with our biomaterials. So this is something that is naturally involved in healing, but is induced even further upon our biomaterial introduction. This shows up in other people's data as well, despite the fact that they didn't actually report it. They just put it into NCBI databases, and I found it. So you can see that this protein shows up in the early stages of healing. It also shows up as the key serpins that are expressed when humans get injured. So you can see serpin B3 and B4, which are B3A duplicates. This is positionally specific. So if you get an injury in the skin, you're going to start making it. But if you bite your tongue, there's already a lot there, but it's not going to really respond to any change. So this is a positional specific expression. We have seen it in our own mouse wounds. So we see that this protein comes up upon injury. We can get human cells to make this in culture by scratching them. And then we can see by doing IHC that this protein shows up not in healthy skin, but rather upon the healing process. You can see that these cells right at the edge of the skin light up and really drive that acceleration. So we ask questions about what is this protein doing, and we can see that when cells overexpress serpin B3, they start to show evidence of this EMT process, and so they're losing their E-cadherin, they're activating phospho-STAT3. Because it's a circulating protein, we added it onto cells, and we can see that even keratinocytes that are exposed to extracellular serpin B3 start to exhibit these same phenotypes. It accelerates scratch wound closure, but it's specific to keratinocytes because fibroblasts don't respond. And so you can see that cells that are exposed end up moving very, very quickly as EGF versus something like a control. In vivo, we see that if we treat this protein like a drug and add it onto wounds, we can get wounds to heal a little bit better. We can see that this protein sticks around even after the skin is healed. So this is indicating this new idea that skin can be primed for subsequent injury. If you have an injury and your skin heals up, it is sort of ready for a certain period of time for subsequent injury. And so this protein sticks around. And something that we're looking into is why, because it's very unusual that keratinocytes are the primary target, is it that when we treat wounds with this protein, they actually heal better, not just faster, but their remodeling is much, much better. The skin looks essentially like healthy skin. So where are we going? What's closest on the horizon? So I recently won this R01 award in collaboration with Kaushal and a couple of other investigators on campus, looking at the mechanistic investigation of this protein in another keratinocyte-involved disease, psoriasis, which is characterized by keratinocyte hyperproliferation. And this should not be so blasted out, but you can get a sense of how much serpin B3 is in healthy skin. It's just at the very, very top layer of the skin. When you have psoriasis, this is brand new data from my PhD student. You can see that when you induce psoriasis in an animal, this protein just blasts out of the skin. And we can try to manipulate that pathway using some experimental drugs to try to suppress that. And so we've seen reductions in clinical evidence of disease. So this is my last slide. So in CBET, we do a lot of biomaterial engineering, but we also run sort of a biological discovery engine on the basis of this. And so we're looking at how biomaterial tissue responses can indicate healthy tissue signaling, which we can then explore fundamental protein function and then look at those proteins as a disease target to loop back on this process itself. So thank you very much for your attention. I'm happy to take any questions or we can talk offline.
Yeah, so any questions? Come on, he's a new faculty member. Give him a hard time. So what is the enzymatic activity that serpins do?
So they're inhibitors. They actually don't have any enzymatic activity on their own.
Right.
So they're sort of sitting around doing nothing until their target becomes active and they act as a negative regulatory element.
Right, and so their name?
Serine protease inhibitor.
Yeah.
So they can inhibit serine and cysteine proteases. They also do some interesting things where they can bind to receptors and just act as signaling molecules. I should mention, so this work is going to appear this week in PNAS. So we're very proud of that. I was hoping it was going to be online today, just so I could be like, and here it is, but sometime this week. So,
Right, right, right. And they were first found where?
where was a serpent first found?
Yeah, weren't they first identified in viruses?
No. I don't know where the first serpent was found. That's a
Okay.
great question. That's a great question. They've been studied for a long time. One of the first crystal structures that was solved was of a serpent by Dr. Huber, who won the Nobel Prize for his work in crystallography. And one of his first structures was a serpent in the 1960s. So they've been around for a while. People have known them for a while, but they've kind of been like held by the crystallographers because it's such an interesting family. And I often find that biologists, like, they've heard of them, like Pi1 or alpha-1 antitrypsin, but they don't really know about the serpent aspects of it. They're a really interesting protein family.
Nice talk, Jordan. Really enjoyed that.
Thanks, Colin.
You brought out the connection between wound healing and cancer. Of course, I'm very interested in that. Any effects either overdriving or inhibiting the serpin B3 and skin cancer?
Yeah, so that's exactly a project that we're starting right now. So we found, and we recently reported in Biomaterials with another pathway that we can inhibit motility by stimulating the histamine pathway in a certain way. And there's some evidence that there may be convergence along with serpins, so especially in squamous cell carcinoma. So this is an active question we're asking: can we manipulate this protein by downregulating it to prevent and sort of arrest the metastatic process?
So we've recently discovered a negative correlation between across species between cancer mortality and trauma mortality, which we think is probably a wound healing trade-off with cancer susceptibility.
Interesting.
Do you have any data on like wound healing rates across species?
Oh, that's fascinating. I don't, but I want to do it now. That's great. That's really interesting.
Great. Any other questions? All right, well, thank you. That was great.
Thank you.